Right. Thanks, Kevin, and thanks, Daria. Thanks for everyone who turned out. This is really a remarkable thing for me because, as Shreet kind of explained a little bit, I was initially a student in the SPARK program. I got to experience it really from a learning perspective, really broadened my education here at Stanford. And now as a member of having a project in the program, I'm really reaping the benefits of what SPARK has to offer. So the program that I'll tell you about today was work that came out of my graduate studies and Brian Koblika's lab, and work that I'm continuing in my own group here at Stanford. The work is largely collaborative, and the people that are involved at Stanford are myself, Brian Koblika, and Gregory Scher. So what's the problem? And just to kind of frame the problem, I like to put this cover of Time up, which is from last year. And as many of you know, there's a remarkably important epidemic of prescription opioids in this country. And it's framed really well by this Time cover, which suggests that these medications, while on the one hand are the most powerful painkillers that we've ever identified, on the flip side, they're creating one of the worst epidemics that the United States has ever seen. Okay, so let's put some numbers to this Time cover. On the one hand, in the US, we have a chronic pain crisis. So the Institute of Medicine estimates that about 1 in 3 Americans, or about 100 million Americans, suffers from chronic pain. And that's defined as pain that lasts over 12 weeks and is not treated by currently available therapeutics. If we put a price tag on that, that costs about $635 billion per year. And to give you some perspective, that's higher than the cost of cancer, diabetes, and heart disease combined. And finally, as many of you might have either observed from personal experience or from others around you, chronic pain ends up being a major cause of missed work and absenteeism. And perhaps because of this large pain crisis, over the past decade or so, the entire medical community has become more comfortable using what are most effective ways of dealing with pain. And one of those medications some of you may be familiar with is oxycodone. So that's on the one side. We have medications to deal with pain, but we're not doing a great job. There's still a lot of people in pain. On the flip side, the use of these medications has created an epidemic. And this is not an epidemic in use of illicit opioids. This is an epidemic in the use of prescription opioids. And here's data for the past 15 years from the National Institutes of Drug Abuse showing that in 2014, there were about 30,000 deaths from all opioids. And the majority of those were actually driven by prescription opioids. Now, if you compare that to drugs, illicit drugs like cocaine, you can see that's pretty flat. So there's really something specific about opioids that's happening over the past 15 years. So how do opiates work, and where do we fit in? So the way that drugs like morphine or codeine work is that they act at opioid receptors in the body. Some of these receptors are in the brain, and these are responsible for mediating painkiller activity or analgesia. There's other receptors that are in the gut, the same receptors as the opioid receptors, and these cause constipation. And over the past 3 or 4 decades, what's become clear is that both the side effects and the beneficial effects of opioids are mediated by a centrally important receptor called the mu-opioid receptor. And perhaps the most important side effect for the opiate receptors and opioids is respiratory depression. And this is what actually kills people in overdoses. That combined with the development of tolerance and addiction creates a self-perpetuating cycle, and that's what we're seeing with this opiate epidemic. Okay, so this is the team. Before I even tell you about our work, I want to highlight the team of people that did this work. There's a number of people at Stanford. We had great collaborators across the world: Peter Gemeiner in Germany, Brian Roth at the University of North Carolina, and Brian Scheuch at UCSF, along with some other advisors who've helped us kind of formulate this into a business plan. So what's the way that we wanted to tackle separating pain killing from side effects? And this comes from some mouse studies that are now about 15 or 20 years old. And the idea was that in arrestin knockout mice, so arrestin is a molecule that interacts with the mu-opioid receptor. And in mice where you remove this arrestin molecule, but you feed them morphine, those mice get really profound pain-killing activity, but they have decreased amounts of side effects like respiratory depression, constipation, and intolerance. So the hope for the field for a long time had been to identify a so-called biased agonist, a drug that would bind to the opiate receptor and then induce selectively interactions with the G protein, GI, and therefore cause analgesia, but somehow not cause interactions with the beta-arrestin molecule and therefore may have decreased side effects. So the approach that we took came out of our work as structural biologists. And in 2012, while I was in Brian Koblika's lab, I determined the first crystal structure of the mu-opioid receptor, in this case bound to an antagonist. And what we did with that crystal structure is we took a large compound, a large virtual library, in this case about 3 million molecules, and screened for whether any of these molecules might bind within that binding pocket of this crystallized structure. And this was all done in silico. And then we took about the top 23 of those molecules, validated them experimentally, and then optimized them by somewhat traditional medicinal chemistry approaches. In less than 50 molecules, we arrived at a molecule called PZM21. And here's PZM21 now docked into the binding pocket of the mu-opioid receptor. And in orange, I'm comparing it to what would be a binding pose for a molecule that looks relatively similar to morphine. And this is a crystallized molecule called BU72. And what I want to point out here is that using these kind of structure-based methods, we were able to find a drug that in some ways recapitulates some of the features of morphine binding, but in some ways is quite different. I won't go into the details of that here. Okay, so what does this molecule actually have some of these interesting features? And this is where we really started to get excited. It turns out that this molecule, PZM21, fulfills some of the criteria of a so-called biased agonist for the mu-opioid receptor. And I'll show you two experiments that really suggest this. On the left is a signaling assay looking at how well the opiate receptor is signaling through the G protein GI. On the right is a signaling assay where we're looking at how well the opiate receptor is coupling to this molecule beta-arrestin. So on the left, what you'll see is in a dose-response mode, on the black curve here is a molecule called DAMGO, which looks a lot like the endorphins that are in your body. And you can see that endorphins or morphine or PZM21, they all activate the G protein pretty well. On the flip side, if you look at what these molecules do to the beta-arrestin, the endogenous endorphins do activate or recruit this beta-arrestin quite well. So does morphine, but we see essentially no activity for this molecule PZM21. Okay, so that's great. That's where we first started to get excited. But if we were correct, and if the field was correct that such a molecule may induce analgesia or pain-killing activity without some of these side effects, the only way to find that out would be in at least an animal model. So we did just that. The first experiment that we did was to first establish whether this molecule actually decreases pain. And if it decreases pain, does it do it as well as morphine? So one of the kind of classical assays in the opiate receptor field is to do what's called a hot plate assay. And the experiment is you take a mouse and you put it on a hot plate, a plate that's about heated to 50 degrees or so. And then you ask whether it shows behaviors consistent with perception of pain. If you give this mouse a drug like morphine, you get an analgesic response that shows dose dependence, and it has a time course consistent with morphine's pharmacokinetics. So they have analgesia that comes on, and then after about 120 minutes, it wears off. When we give mice this new compound PZM21, we again get analgesia, suggesting that this molecule does reduce pain, comparable to morphine. It's dose dependent, and the experiments I'm not showing you here, we know that this effect comes from the mu-opioid receptor because this effect is gone in mu-opioid receptor knockout mice. Okay, so now we have a molecule that we believe has pain-killing activity that's novel, but what about some of the side effects that I told you about earlier? The first experiment that we did was to examine whether PZM21 suppresses breathing, and this is really important because this is the dose-limiting side effect for opiates. This is, again, what causes opiate overdoses. So this is an experiment where we're basically very accurately measuring the breathing rate of a given mouse after giving it a drug. So if you give the mouse morphine, you can observe there's a profound decrease in the breathing rate. If we give the same mice PZM21, we observe basically no difference compared to vehicle. And this is really profound because there's essentially no other opiates for which this has been observed. And PZM21 therefore seems to be among a new class of opiates that really seems to start separating pain-killing activity from respiratory depression. Now, I'm going to turn to a part of the story where where we didn't quite anticipate this, but we're even more excited. And that is that there's some preliminary data that we have that suggests that PZM21 may not be addictive. And I'll show you two experiments that kind of suggest this. The first is an open field locomotion assay. And in such an experiment, you put a mouse in an apparatus like this where you can really accurately measure its movements. And drugs that turn on reward pathways like cocaine, amphetamines, or morphine cause these mice to move around this cage quite a bit. So we tested what PZM21 would do in such an experiment. And we did this experiment, when you give the mice morphine, you see that they start moving around quite a bit, and that resolves after about 120 minutes. When we give the mice PZM21, we see no effect. And this was our first kind of hint that perhaps PZM21 doesn't activate the same reward pathways that other opiates do. Now, a better experiment for looking at addiction or looking at rewarding properties is a conditioned place preference experiment. And in this kind of experiment, there are two chambers that are connected by a little gate in the middle, and each chamber is very similar but slightly differently patterned. And you do a classical conditioning experiment. You pair one of the chambers with the drug and the other chamber with the vehicle, and you see if there's if the mouse finds a drug rewarding, it'll basically want to spend more time in the chamber that's paired with a rewarding drug. So okay, on day one, we just let the mice freely explore both chambers for both, either for the morphine-treated group or the PZM21 group. They basically spend equal amounts of time in each chamber. After about 10 days of conditioning, where we pair one of these chambers with drug and the other with vehicle, what we see is that for morphine, the mice really like to spend time in the morphine-paired chamber, but for PZM21, we don't see that effect. And that's really our most exciting evidence that PZM21 may not have the addictive properties of previously known opioids. So this was, again, one of these great examples of an academically driven project where after the academics had been done, we really didn't have a great framework on how to go forward and actually create meaningful benefit for patients. And I'm putting up this Gantt chart, not for the specifics, but just to show you that this kind of organizational thinking that's critical to moving things forward into the clinic is something that academics often lack. And just putting this Gantt chart together for me at least helped me formulate what we're going to do. This is an ongoing SPARK project, and we're right about year 1, quarter 4 right here. I'll give you one piece of data that SPARK enabled that I think is extremely exciting, and that's just the preliminary pharmacokinetics of PZM21, something that we could really not do with the academic funding or the academic work. And what these studies has showed, what basically showed us is that PZM21 has a 50% oral bioavailability in rats. And if you think about what this means for attractiveness to an investor, this is a game changer. Now we're talking about an orally bioavailable opiate for chronic use that separates analgesia or pain-killing activity from side effects. And this is the kind of value that I think SPARK really brings to academic investigators like myself. So I want to end with acknowledgments. The work that I described was done, again, over a large number of people over many different labs across the world, both in Brian Koblika's lab here, Brian Schweikert's lab at UCSF, the Roth lab at UNC, Peter Gemeiner's lab in Germany, and Greg Scher's lab here at Stanford. I can't thank the SPARK program enough, not just for this specific project, but just for the amount of learning and realism that I've been able to gain over the past 5 or 6 years that I've been coming every Wednesday afternoon. And then, of course, the numerous SPARK advisors who've offered help on this project, but just perspective and guidance on many different things. So thanks again, and thank you for your time.
So again, we have time for a couple of questions. All right, terrific. Oh, got one.
Again, still overlapping with us. So I have a question. So sometimes the drug, anti-painkiller drug works in rodent, but not work in primate. Have you tried have you tested the drug in primate?
We haven't yet. I would say, though, that for opiate agonists, on average, the on-target liability is relatively low. So I think if you're going for a new pain pathway, that might be true. But I think almost every opiate that's known to be bioactive in humans or primates has been, at least to my knowledge, is efficacious in these pain assays in mice. There's a pretty decent correlation, at least from our perspective.
Okay. So
we have not done the primate studies, though. We'd love to, yeah.
The 3D model come from the human, right?
So that specific crystal structure is of the mouse mu-opioid receptor, and there are basically no differences. There's not a single amino acid in the binding pocket that's different.
Okay, thanks.
Yep. Yeah.
Just a very simple question, which is, so does naloxone reverse the activity? of this?
Absolutely, yeah.
Ashish, thank you very much. That was great.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.